Continuing with the chapter that will stop right now, we have the first recombinant growth hormone, which is called somatotropin or protropin. It was produced and marketed by Genentech beginning in 1985. It had an amino acid sequence that was identical to that of human growth hormone, except that there was an extra methionine residue at the N-terminal end of the peptide chain, which was thought to prolong its half-life. It was discontinued in the late 1900s. Treatment of children with human growth hormone typically entails daily injections during the years when the child is growing. The cost of the treatment varies depending on the country and size of the child, but is generally approximately $10,000 to $30,000 per year. In addition, in 2004, the US FDA approved the use of recombinant human growth hormone for individuals whose short stature was a cause for concern. Was caused by a variety of medical conditions other than a human growth hormone deficiency. The strategy of designing a protein by either functional domain shuffling or directed mutagenesis can be used to augment or constrain its mode of action. For example, native human growth hormone binds to both growth hormone and prolactin receptors that occur on a number of different cell types to avoid unwanted side effects. During therapy, it is desirable that the human growth hormone bind only to growth hormone receptors. Because of the segment of the growth hormone molecule that binds to the growth hormone receptor overlaps but is not identical to the portion of the. This is a schematic representation of that HSA gene, IFN alpha gene, right? And the molecule that binds to the prolactin receptor, it should be possible to selectively decrease the binding to the prolactin receptor. Site-specific mutagenesis of the clone human growth hormone cDNA was used to change some of the amino acid chains that act as ligands for zinc 2 because the iron is required for the high affinity binding of human growth hormone to the prolactin receptor. As hoped, these modifications yielded human growth hormone derivatives that bound to the growth hormone receptor but not to the prolactin receptor. These derivatives are being tested for safety and efficacy in humans. This is a schematic representation of native and modified youth hormone factor oligonucleotide-directed mutagenesis was used to alter human growth hormone so that it no longer bound to the prolactin receptor but retained its specificity for the growth hormone receptor. And now, as a consequence of its relatively short half-life span in plasma, human growth hormone therapeutic currently requires subcutaneous injection once a day. This treatment is both inconvenient and expensive. Therefore, it would be advantageous to have a long-lasting form of human growth hormone to this end. The extracellular domain of the human growth hormone receptor was fused to human growth hormone using a 20 amino acid long-linked linker peptide consisting of 4 repeats of the amino acids. This construct has a very strong tendency to dimerize as the growth hormone moiety, where one molecule binds with the receptor portion of another molecule. When the growth hormone construct was tested in rats, a single injection promoted growth for 10 days compared to the usual requirement in rats for daily injections. It is thought that the dimerization of the growth hormone construct stabilizes human growth hormone in vivus so that it is cleared from plasma approximately 300 times more slowly than free growth hormone. Under these conditions, the active monomeric form it is slowly released from the inactive dimeric growth hormone, allowing it to bind to the growth hormone receptor. This experiment is certainly intriguing. It remains to be determined whether humans respond in a similar manner to the dimerized complex. Another method that has been devised to prolong the active lifetime of human growth hormone includes fusing the coding sequences for the C-terminal end of human growth hormone to 22 kilodaltons with the N-terminal end of human serum albumin 67 kilodaltons. This fusion protein is called albutropin. It has a molecular mass of 89 kilodalton and is produced by a strain of yeast that has been genetically modified so that the proteins that it produces have a similar number of post-translational modifications. The stabilization of the human growth hormone portion of albutropin reflects the stability of human serum albumin, which has a half-life in serum of about 19 days. Albutropin has been shown to be effective in both cases. This is a schematic is a representation of de-derivatization of growth hormone by coupling it to a portion of the growth hormone receptor using a 20 amino acid peptide. Monomeric derivative, dimeric derivative, and monomeric derivative bound to the growth hormone receptor. Take screenshots, please. And rats and monkeys in which high level of the protein in serum were observed 5 days after it was administered. Moreover, albutropin has been successfully phase 1 clinical trials. Tumor necrosis factor alpha. While a number of these studies have clearly shown that tumor necrosis factor alpha TNF alpha is a potent anti-tumor agent. If TNF alpha, before that, tumor necrosis for alpha. While a number of studies have clearly shown that tumor necrosis factor alpha, TNF alpha, is a potent anti-tumor agent, it has not been widely used in this capacity because of its severe toxicity. If TNF alpha could be delivered directly to the site of action, that is the tumor, then lower doses could be used and the unwanted side effects would be diminished. To develop a version of TNF alpha with tumor specificity, DNA encoding the peptide cis, peptide cis, ASN, GLI, etc., which targets a tumor cell surface protein, was fused to TNF alpha DNA. The fusion protein contains 6 amino acid extension at its N-terminal end. In mice, the cytotoxic activities of cis ASN at this particular codon, activity of this particular peptide and TNF alpha were identical, indicating that the additional amino acid did not prevent protein folding, combining of 3 subunits to form a trimer or binding to receptors. However, the modified version of TNF alpha was 12 to 15 times more effective at inhibiting tumor growth than the unmodified form. Moreover, a higher percentage of mice with lymphoma survived after treatment with the modified factor. In addition, all the mice that were treated with the modified factor and survived for 30 days, survived the second and third challenge with mouse lymphoma cells. These data indicate that there is a significant benefit, at least in mice, to fusing TNF alpha with a short targeting peptide. Nevertheless, this work must be regarded as a preliminary until its efficacy is demonstrated in humans. Enzymes, DNA is one. Cystic fibrosis is one of the most common fatal hereditary diseases among Europeans and their descendants, with approximately 30,000 diagnosed cases in the United States and another 23,000 cases in Canada and Europe. It is estimated that a mutant cystic fibrosis gene is carried by 1 in 29 Europeans, 1 in 65 African Americans, and 1 in 150 Asians. Individuals with cystic fibrosis are highly susceptible to bacterial infections in their lungs. Antibiotic treatment in patients who have these recurring infections eventually leads to the selection of antibiotic-resistant bacteria. The presence of bacteria, some alive and some live, contributes to the accumulation of thick mucus in the lungs. These patients making breathing very difficult and acting as a source of further infection. The thick mucus in the lungs is a result of the combination of the alginate that is secreted by the living bacteria, the DNA that is released from live bacterial cells, and degenerated leukocytes that accumulate in response to the infection, as well as filamentous actin derived from cytoskeletons of damaged epithelial cells. To address this problem, scientists at the US biotechnology company Genentech isolated the gene for the human enzyme deoxyribonuclease 1, DNase 1, and subsequently expressed the gene in Chinese hamster ovary, CHO cells, and in culture. DNase 1 can hydrolyze long polymeric DNA chains into much shorter oligonucleotides. The purified polymeric DNA chains into much shorter oligonucleotides. The purified enzyme was delivered in aerosol mist to the lungs of the patient with cystic fibrosis. The DNase 1 decreased the viscosity of the density of the mucus in the lungs, made it easier for those patients to breathe. While this treatment is not a cure for cystic fibrosis, it nevertheless relieves the most severe symptom of the disease in most patients. Enzyme was approved for use by the FDA in 2024. It had sales of approximately $100 million in 2000. It's so hot. The monomeric form of actin binds very tightly to DNase 1 inhibitor constant KIL-1NM and inhibits its ability to cleave DNA. This interaction limits the effectiveness of DNase 1 as a therapeutic agent. On the basis of X-ray crystallographic studies, it was possible to predict which amino acid residues of DNase 1 interact with actin and were therefore possible targets for change by direct immunogenesis. For example, changing amino acid 144 from alanine to arginine or amino acid 65 from tyrosine to arginine decreased the binding of DNase 1 to ACT up to 10,000-fold. In addition, the actin resistance mutants had 10 to 50 folds more DNase 1 activity than the native enzyme. It is not known whether any additional benefit might be realized by combining the amino acid changes from several actin-resistant mutants. The clinical efficacy of DNase 1 mutant enzyme that does not bind actin still remains to be demonstrated. Alginate lyase. Alginate is a polysaccharide polymer that is produced by a wide range of seaweeds and both soil and marine bacteria. Alginate is composed of chains of the sugars beta-D-menulronate and alpha-1-glucuronate. Alpha-1-glucuronate. The properties of a particular alginate depend on the relative amounts and distribution of these two saccharides. For example, stretches of alpha-1-glucuronate residues form both interchain and intrachain cross-links by binding calcium ions, and the beta-D-menulronate residues bind at the metal ions. The cross-linked alginate polymer forms an elastic gel. In general, the structure of an alginate. We'll come to all these representations later. Innate polymer is related to its viscosity, which is in turn directly proportional to its molecular size. The excretion of alginate by mucoid strains of Pseudomonas aeruginosa that infect the lungs of patients with cystic fibrosis significantly contributes to the viscosity of the mucus in the airways. Once mucoid strains of P. aeruginosa have become established in the lungs of cystic fibrosis patients, it is almost impossible to eliminate them by antibiotic treatment. This is because the bacteria form biofilms in which the alginate prevents the antibiotics from coming into contact with the bacterial cells. One experiment was shown that the addition of alginate lyase, which can liquefy bacterial alginate together with or prior to antibiotic treatment, significantly decreased the number of bacteria found in biofilms. Those results suggest that in addition to the DNase 1 treatment, depolymerization of the alginate will help clear blocked airways of individuals with cystic fibrosis. An alginate lyase gene has been isolated from a Flavobacterium species, a gram-negative soil bacterium that has a strong reducer of this enzyme. A Flavobacterium clone bank was constructed in E. coli and screened for alginate lyase producing clones by plating the anti-clone bank on the solid medium containing alginate. Following growth, colonies that produce alginate lyase formed a halo around the colony when calcium was added to the plate. In the presence of calcium, all of the alginate in the medium except in the immediate vicinity of an alginate lyase positive clone becomes cross-linked and opaque. Since hydrolyzed alginate chains do not form cross-links, the medium surrounding an alginate lyase positive clone is transparent. Analysis of a cloned DNA fragment from one of the positive colonies revealed an open reading frame encoding a polypeptide with a molecular mass of approximately 69,000 daltons. Detailed biochemical studies and genetic studies have indicated that this polypeptide is a precursor of the three different alginate lyases produced by the Flavobacterium sp. After the 69,000 dalton precursor is produced, the proteolytic enzyme cleaves of an N-terminal peptide of about 6,000 daltons. The 63,000 dalton protein can lyse both bacterial and seaweed alginates. Cleavage of the 63,000 dalton protein yields a 23,000 dalton enzyme that depolymerizes seaweed alginate into a 40,000 dalton enzyme that is effective against bacterial alginate. To produce large amounts of the 40,000 dalton enzyme, the DNA corresponding to the enzyme was amplified by the polymerase chain reaction PCR and then inserted into Bacillus subtilis plasmid vector fused to a Bacillus subtilis alpha amylase leader peptide to direct the secretion of the protein, and placed under the transcriptional control of a penicillinase gene promoter. Transformation of beta subtilis cells with this construct yielded colonies with large halos and solid medium containing alginate after calcium was added. When these transformants were grown in liquid medium, the recombinant algae layers were secreted into the culture broth. Further tests show that the enzyme efficiently liquefied alginates that were produced by mucoid strains of P. aeruginosa that have been isolated from the lungs of patients with cystic fibrosis. Additional studies are necessary to determine whether recombinant alginate lyase is an effective therapeutic agent. And now on to phenylaniline ammonia lyase. The human genetic disease phenylketonuria resulted from the impaired functioning of the enzyme phenylaniline hydroxylase. In the US, approximately 1 of every 12,000 newborns has phenylketonuria. When phenylaniline hydroxylase, which oxidizes phenylaniline to tyrosine, is deficient, the normal cognitive development of an individual is impaired, and mental retardation ensues to build-up of phenylaniline. Following diagnosis of phenylketonuria, either prenatally or shortly after birth, the treatment entails a controlled semi-synthetic diet with low levels of phenylaniline through infancy and possibly for life. A possible alternative treatment would be the administration of the enzyme phenylaniline hydroxylase. Unfortunately, the phenylaniline hydroxylase is a multi-enzyme complex that is not very stable and requires a cofactor for activity. On the other hand, phenylaniline ammonia lyase, which converts phenylaniline to ammonia and tranexamic acid, is a stable enzyme that does not require a cofactor and could potentially prevent the accumulation of phenylaniline in phenylketonuria patients. To test this concept, the gene for phenylaniline ammonia lyase from the yeast Rhodosporidium pteryloides was cloned and overexpressed in E. coli. Preclinical studies were conducted with the mice that were defective in producing phenylaniline ammonia lyase and therefore accumulated phenylaniline. With these mice, plasma phenylaniline levels were lowered when phenylaniline ammonia lyase was injected intravenously or encapsulated enzyme was administered orally. Thus, at least in mice, phenylaniline ammonia lyase is an effective substitute for phenylaniline hydroxylase, and the orally delivered enzyme is sufficiently stable to survive the mouse gastrointestinal tract and still function. Although this report is preliminary, a combination of oral enzyme therapy with phenylaniline ammonia lyase and a less stringent low phenylaniline diet might serve to improve the quality of life of individuals affected with this disease. Alpha antitrypsin. The processing of a number of different pathogenic bacterial or viral precursor proteins by human proteases occurs when the protease recognizes the amino acid sequence with peptide-bound cleavage on the C-terminal side of the C-terminal arc as indicated by the arrow, where X is any of the 20 common amino acids. Since this processing step is common to several infectious agents, a therapeutic agent that targeted the processing enzyme and blocked its activity might act as a broad-spectrum antibacterial and antiviral agent. When a variant of human alpha-1 antitrypsin was genetically engineered and tested in tissue culture experiments, the protein blocked the processing of human immunodeficiency virus, HIV type 1 glycoprotein GP160, as well as measles virus protein F0', and consequently in both cases the production of infectious viruses. When the alpha-1 antitrypsin variant was added to cell cultures, it blocked the production of human cytomegalovirus, a major cause of illness and death in organ transplant recipients and AIDS patients. The alpha-1 antitrypsin variant is both potent and selective against human cytomegalovirus. It is at least tenfold more effective than any current viral inhibitory agent. Its efficacy has been demonstrated in cell culture, but it remains to be determine if the strategy is effective with whole animals. So we're talking of alpha-1 antitrypsin, phenylalanine hydroxylase, phenylalanine ammonylase from phenylalanine, so tyrosine, ammonia, and tranexamic acid. Inactive precursor antitrypsin, proteolytic cleavage active protein. Schematic representation of alpha-1 antitrypsin inhibiting the proteolytic cleavage of pathogenic precursor proteins for human proteases. Now we're talking of glycosidases. The ABO blood group system is based upon the presence or absence of specific carbohydrate residues on the surface of erythrocytes, endothelial cells, and some epithelial cells. The monosaccharide that determines blood group A is a terminal alpha-1,3 linked N-acetyl-galactosamine, while the corresponding monosaccharide of blood group B is alpha-1,3 linked galactose. Group O cells lack both of these monosaccharides at the ends of the oligosaccharide chains, and instead of contains alpha-1,2 linked fructose, which is designated the H antigen. Plasma from blood group A individuals contains antibodies against the B antigen. Blood group B individuals have antibodies against the A antigen. And the blood group O individuals have antibodies against both A and B antigens. In practice, this means that individuals with either anti-A or anti-B bodies cannot safely receive a blood transfusion containing incompatible antigen, since this is likely to cause a severe immune response. As a consequence, blood group AB individuals are said to be universal recipients, with those from blood group O are universal donors. Thus, when a blood transfusion is required, it is advantageous to have a large supply of plasma that is from blood group O. Example, in an emergency situation, there may not be sufficient time to check a patient's blood group. Fortunately, digestion of blood cells from either type A or B with specific glycosidases can cause types AB and AB to be converted into type O. But with that, fortunately, digestion of blood cells from either type A or B with specific glycosidases can cause types AB and AB to be converted into type O. These enzymes were found following an extensive screening process of 2,500 fungal and bacterial isolates. Eventually, an active alpha and acetyl-galactosamidase, which converts group A to group O, was found in the gram-negative bacterium Elisabethinia meningosepticum, and one with alpha-galactosidase, which converts group B to group O, was found in Bacteroides fragilis, also in the gram-negative bacterium. The genes were isolated and the proteins were characterized. Both of the enzymes have high specificity for claiming the appropriate monosaccharide under conditions that maintain the integrity and functioning of the treated red blood cells. Moreover, each enzyme could readily be removed from the treated red blood cells following treatment. While this is a very recent and still preliminary experiment, if this novel approach works effectively in a clinical setting, then it should become a boon for all types of blood transfusions. Lactic acid bacteria. Lactic acid bacteria are widely used in the production and preservation of fermented foods, and many have been given the designation generally regarded as safe. Within the food industry, many of these organisms are members of the endogenous microflora of the human gut and have been recognized for their health-promoting properties. Some strains of lactic acid bacteria, notably lactobacilli, are used in probiotic products. A probiotic is a live microorganism that is claimed to confer a health benefit by altering the endogenous microflora of the intestinal tract. Lactic acid bacteria have also been used to treat several gastrointestinal disorders, including lactose intolerance, traveler's diarrhea, antibiotic-associated diarrhea, infections caused by various bacterial and viral pathogens, and immunopathological disorders such as chronic disease and ulcerative colitis. In the past few years, lactic acid bacteria have been used as a host system to express various foreign genes with the idea that these bacteria facilitate the delivery of the proteins encoded by the genes to the human gut. In particular, Lactococcus lactis has been developed as a host for this purpose. L-lactis is a non-pathogenic, non-invasive, non-colonizing, gram-positive bacterium that is often used in the production of fermented foods. Moreover, L-lactis has been used for many years as a human probiotic. Interleukin-10 is the next. Ulcerative colitis and Crohn's disease, both diseases of the intestinal tract, appear approximately 1 in every 500 to 1,000 people in the developed countries of the world. Ulcerative colitis is associated with the excess type 2 T helper cell cytokines, including interleukin-4 and the interleukin-5, whereas in Crohn's disease, type 1 T helper cell cytokines, including TNF-alpha and interferon-alpha and interleukin-2, are overproduced. The treatment for Crohn's disease often includes trying to lower the levels of cytokines, especially TNF-alpha. One approach has been the administration of antibodies against TNF-alpha. Other workers have targeted interleukin-10 as a means of controlling Crohn's disease because it modulates at regulatory T cells that control inflammatory responses to intestinal antigens. However, interleukin-10 is not clinically acceptable because it needs to be administered by the frequent injections or rectal enemas. To overcome this problem, the bacterium L-lactis was engineered to synthesize and secrete interleukin-10. Okay. Experiments were performed with mice to test whether interleukin-10 secreting L-lactis could be used to treat inflammatory bowel disease. First, interleukin-10 secreting L-lactis was fed to mice with ulcerative colitis that are made induced with 5% dextrin sulfate in their drinking water. Second, strains of mice that are genetically incapable of synthesizing interleukin-10 and provide an animal model for ulcerative colitis were tested. In both these cases, the engineered L-lactis significantly alleviated the symptoms of the disease, establishing that this approach works in principle. However, most models for inflammatory bowel disease are not identical to the disease in humans, and a large number of questions remain before the treatment is used with humans. One concern about the use of an interleukin-10 secreting L-lactis strain as a therapeutic approach is the possibility that the genetically modified bacterium will be released to the environment. If this were to happen, the plasmid carrying the interleukin-10 gene and any plasmid-borne antibiotic-resistant marker gene could be spread to the other bacteria in the environment. To prevent this from occurring, a synthetic human interleukin-10 that replaced the L-lactis thymidylate synthase gene Thy-A, which is essential for the growth of the bacterium, was inserted into the bacterial chromosome of L-lactis by homologous recombination. The strain produced interleukin-10 and grew well in the laboratory when either thymidine or thymine was added to the medium. However, when it was deprived of thymidine and thymine, the viability of the bacterium declined by several orders of magnitude. When this modified bacterium was tested on pigs, whose digestive tract is similar to that of humans, it thrived and actively produced interleukin-10. In addition, laboratory experiments demonstrated that the modified L-lactis was extremely unlikely to acquire a thymidylate synthase gene from other bacteria in the environment, confirming both the safety and efficacy of this approach. Recently, clinical trials with this L-lactis strain were initiated. To date, 10 patients with Crohn's disease have been treated. So far, a significant decrease in disease activity has been observed and with only minor adverse events. Moreover, bacteria isolated from patients' feces were not able to grow without the addition of thymidine. In other words, the engineered L-lactis did not acquire a thymidylate synthase gene, indicating that the containment strategy was effective. Thus, the initial indications are that this strategy appears to be working in all humans, as did with the small animals. The next is leptin. It's been estimated that approximately 30% of North American and 20% of the European populations were overweight. Moreover, North Americans annually spend tens of billions of dollars in various weight reduction schemes, most of which are unsuccessful. However, real weight reduction may be obtained by administration, of the protein leptin. Leptin is the product of the OB gene. It's a 167 amino acid protein with a molecular mass of approximately 16 kilodalton. Leptin is synthesized as a precursor with a 21 amino acid long signal peptide that is removed when leptin is secreted. Treatment with recombinant leptin can reduce food intake and correct metabolic perturbations in homozygous leptin deficient mice. Leptin also helps to overcome human congenital leptin deficiency. However, when it is introduced subcutaneously, leptin is not particularly effective in obese patients unless their serum leptin concentrations reach levels 20 to 30-fold higher than normal. This response has been attributed to the inefficient transport of leptin across the blood-brain barrier. To overcome this problem, a scheme for the intranasal delivery of leptin has been devised. When leptin is produced in E. coli, it typically forms insoluble inclusion bodies that must be solubilized and renatured before the active protein is generated. This is a time-consuming, inefficient, and expensive process. One study, the 462 pp cDNA for human leptin without its signal peptide was cloned and expressed under control of the nisin promoter and L-lactase. Nisin is a 34 amino acid residue polycyclic peptide that has antibacterial activity and is used as a food preservative. In L-lactase, leptin was produced efficiently without the formation of an inclusion body and was secreted from the recombinant bacteria. Intranasal administration of the leptin-producing L-lactase strain significantly reduced food intake and body weight in obese mice. This approach opens up the possibility that if delivered properly, leptin might act as an effective weight loss treatment in humans. An HIV inhibitor. Worldwide, the predominant mode of HIV transmission is by heterosexual contact. One possible way to protect women, who currently comprise half of all new cases of HIV/AIDS against HIV infection, is a topical microbicide delivered by a live vaginal lactobacillus strain that prevents HIV infection directly at mucosal surfaces. This strategy seems reasonable because naturally occurring vaginal lactobacillus strains play a protective role in preventing urogenital infections. The compound cyanovirin N, isolated from the cyanobacterium Nostoc ellipsosporum, blocks several types of HIV infection. Nostoc ellipsosporum blocks several steps of HIV infection, preventing virus entry into human cells. Consequently, cyanovirin N is a candidate for a topical microbicide to prevent HIV infections. To ensure this cyanovirin N would be expressed at a sufficiently high level in a vaginal strain of Lactobacillus jensenii, the gene was chemically synthesized to reflect the codon usage found in the bacterium. Typically, the GC content of lactobacilli is about 36%. In addition, during this synthesis of the gene, proline 51 is replaced by a glycine residue. To stabilize the cyanovirin N and 4 amino acids were added to the N-terminus to ensure proper cleavage of the signal sequence. The modified cyanovirin N gene was fused to a strong and constitutive lactobacillus promoter. The final construct was integrated into the chromosomal DNA of a strain of L. jensenii, and when was tested for efficacy, was found to be highly effective at preventing HIV infection in mice. Under these conditions, about 4 micrograms of cyanovirin N per ml were released in the culture medium, monoclonal antibodies. About 100 years ago, horses were inoculated with the bacterium Corynebacterium diphtheriae, which causes diphtheria in humans. The resulting crude horse antiserum was used to treat often fatal childhood disease. In those days, mortality sometimes reached 45%. C. diphtheria infects the throat or tonsils and produces an exotoxin that is lethal to human cells. This exotoxin enters the bloodstream and damages organs that are distant from the primary site of infection. The administration of horse antiserum containing antibodies against the exotoxin provided passive immunity, protecting the patient from a fatal outcome, but the antiserum was given within the first few days after the onset of infection. Unfortunately, this kind of antibody therapy carries considerable risk and is not widely used today. Patients often develop antibodies against the foreign proteins of either whole or partially purified horse antiserum. After a second treatment, the sensitized patient may go back into anaphylactic shock and die. As a result, the use of antibodies as therapeutic agents was considered too dangerous for patients and was used only rarely. However, with the development of hybridoma technology, antibodies are once again seen as potential therapeutic agents. So, hybridoma technology. Hybridoma technology has antibodies that are once again seen as potential therapeutic agents. One reason for the renewed interest in therapeutic antibodies is that it is now possible to engineer antibodies with greatly reduced levels of immunogenicity in humans. In addition, this technique can be used to maintain a continuous supply of pure monospecific antibody. However, the problems of cross-reactivity leading to an immune response and anaphylaxis have not been completely overcome. Thus, the recipient might still produce antibodies to a monoclonal antibody that carries mouse murine determinants. To avoid this problem, human monoclonal antibodies with both specific immunotherapeutic properties and lowered potential for immunogenicity have been produced. In fact, a number of monoclonal antibodies have been approved for treating human diseases. Structure and function of antibodies. An antibody molecule, immunoglobulin, consists of two identical light L protein chains and two identical heavy H protein chains held together by both hydrogen bonding and precisely localized disulfide linkages. The N-terminal regions of the L and H chains together form the antigen recognition site of each antibody. Antibody genes can be readily manipulated because the various functions of an antibody molecule are confined to discrete domains or regions. The sites that recognize and bind antigens consist of three complementary determining regions: CDRs that lie, we'll come back to these note later, within the variable V14, VH, and V1 regions at the N-terminal ends of the two H and 2L chains. The CDRs are the part of an antibody molecule with greatest variability in amino acid sequence. In addition to the variable regions, each L chain contains one constant region or one domain C1, and each H chain has three constant regions or domains CH1, CH2, CH3. When antibodies are digested with the proteolytic enzyme papain, three fragments are released, two identical Fab fragments, each of which contains an intact L chain linked by a disulfide bond to the CH1 region of the H chain and one Fc fragment, which consists of the two H chain fragments, each containing CH2 and CH3 domains, and joined by a disulfide bond. The Fab fragment retains the antigen binding activity. In fact, the N-terminal half of the fragment, which is called the Fv fragment, contains all the antigen binding activity of the intact antibody molecule. The amino acid sequence of this portion of the antibody varies considerably from one molecule to another. Each of the constant and variable regions consists of approximately 110 amino acid residues. A complete antibody molecule has a molecular mass approximately 150 kilodaltons. A Fab fragment is around 50 kilodaltons, and an Fv fragment is about 25 kilodaltons. In an intact antibody molecule, the Fc portion elicits several immunological responses after antigen-antibody binding occurs. The complement cascade is activated. The components of this system break down all cell membranes, activating phagocytes and channel signals to mobilize other components of the immunological response system. An antibody-dependent cell-mediated cytotoxicity, it's ADCC, which is the result of the binding of the Fc portion of the antibody. An Fc receptor of an ADCC effective cell is produced. The bound effector cell releases substances that lies the foreign cell to which the Fab portion of the antibody molecule is bound. After the Fab region binds to a soluble antigen, the Fc portion of an antibody can be bound to Fc receptors of phagocytic cells, which engulf and destroy the antibody-antigen complex, preventing rejection of transplanted organs. Now, in the 1970s, passive immunization was reconsidered as a way of preventing immunological rejection of a transplanted organ. The rationale was to administer to patients a specific antibody that would bind to certain lymphocytes and diminish the immune response directed against the transplanted organ. The mouse monoclonal antibody OKT3 was approved. In 1986, the FDA, for use as an immunosuppressive agent for after organ transplantations in humans, table 10.3, lymphocytes that differentiate in the thymus are called T cells. Various members of the T cell population act as immunologic helper and effective cells and responsible for organ rejection. The OKD3 monoclonal antibody binds to a cell surface receptor called CD3, which is present on all T cells. As a result of all, full immunological response is blocked, and the transplanted organ is not rejected. Immunosuppression by means was reasonably effective, although as anticipated, because the antibody was from a mouse, there were some side effects including fever and rash formation. Recombinant antibodies. Hybrid human mouse monoclonal antibodies. The modular nature of antibody functions has made it possible to convert a mouse monoclonal antibody into one that has some human segments but still retains its original antibody antigen binding specificity. This hybrid molecule is called a chimeric antibody, or with more human sequences, a humanized antibody. The difference between a chimeric and a humanized mouse monoclonal antibody depends on which portions of the mouse antibody are removed. The first portion of a mouse monoclonal antibody that was targeted for replacement with a human sequence was the mouse Fc fragment. The mouse Fc fragment was chosen because it functions poorly as an effector of immunological responses in humans. It is also the most likely fragment to elicit the production of human antibodies, to diminish immunogenicity, and to introduce human Fc receptor effector capabilities. The DNA coding sequences for the Fe regions of both the L and H chains of the human hemoglobin were substituted for the Fe DNA sequences for the L and H chains from a specific mouse monoclonal antibody. This replacement of Fe coding genes can be accomplished by using either oligonucleotides with in vitro DNA replications or cloned DNA segments. The DNA constructs for both chimeric chains were cloned into an expression vector and transfected into cultured B lymphocytes from which the chimeric antibody was collected. Chimeric antibodies are composed of approximately 70% human and 30% mouse DNA sequences. When a chimeric antibody that contains a binding site from a mouse monoclonal antibody directly against the surfaces of human colon cancer cells or intestine patients with colorectal cancer, it remained in the blood system about 6 times longer than the completed mouse monoclonal antibody, thereby extending the period of effectiveness. Only one patient of the 10 developed a mild immunological reaction to the chimeric antibody. However, in this trial, no anti-tumor effects were observed, an outcome that may be due to the low dose levels to the advanced stage of the cancer in the subject. The humanizing of mouse and rat monoclonal antibodies has been taken one step further than the formation of chimeric molecules by substituting into human antibodies, only the CDRs of the rodent monoclonal antibodies. Humanized antibodies consist of approximately 95% human, 5% mouse DNA sequences. Because these engineered human antibodies have antigen binding affinities similar to those of the original rodent monoclonal antibodies, they may be more effective therapeutic agents. Humanizing of rodent monoclonal antibodies may be performed as follows, starting with the rodent hybridoma cell line cDNAs. For the LNH chains are isolated, the variable region of these cDNAs are amplified by PCR. The oligonucleotide primers that are used for this amplification are complementary to the sequences at the 5' and 3' ends of the DNA encoding the variable genes from the nucleotide sequences of the cDNAs for the LNH regions VL and VH. It is possible to delineate the limits of the CDRs. It is usually straightforward to determine where the CDRs begin and end, because these regions are highly variable in sequence, while the sequences of the framework regions FRs are relatively conserved. On the basis of the sequences of the DNAs encoding the rodent CDRs, 6 pairs of oligonucleotides PCR primers are synthesized. Each pair of primers is designed to initiate the synthesis of the DNA for one of the 6 rodent CDRs, 3 from the L chain and 3 from the H chain. In addition, each primer includes an extra 12 nucleotides at its 5' end, complementary to the flanking regions within the human framework DNA into which the DNA for the rodent CDRs is targeted. Oligonucleotide-directed mutagenesis is then used to replace, one at a time, the complete DNA sequence for each of the human CDRs with the amplified DNA for the rodent CDRs. Thus, it is necessary to carry out 6 cycles of oligonucleotide-directed mutagenesis, 1 cycle to replace each CDR. This procedure in effect grafts the rodent CDRs into human antibody framework. The humanized variable region cDNAs are then cloned into expression vectors, which are then introduced into appropriate host cells, usually either E. coli or mammalian cells, for the production of antibodies. To date, more than 50 different monoclonal antibodies have been humanized. While this technology is clearly effective and widely applicable, it is nevertheless time-consuming and expensive. Probably as described below, in the future, other strategies will be used to produce human antibodies and antibody fragments, such as 1-phage display combinatorial libraries that are constructed from mRNA from human B cells, from non-immunized donors, and 2 transgenic mice that express the entire human antibody repertoire. Human monoclonal antibodies. Although most of the immunotherapeutic agents that have been developed have been effective, there are drawbacks to the use of monoclonal antibodies that contain non-human sequences. For example, if multiple treatments are required, which is often the case, it is desirable that the antibody contain no or only a very limited amount of non-human sequences to prevent immunological cross-reactivity and sensitization of the patient. Unfortunately, it is very difficult to create human monoclonal antibodies for a number of reasons. The human chromosome of fused human lymphocyte mouse myeloma cells during hybridoma formation are unstable, and cells that produce a human monoclonal antibody are rarely formed. To date, no human myeloma cell line has been discovered that can replace the mouse myeloma cell line in the procedure. Even if it were possible to form human hybridoma cell lines, it is contrary to accept norms of medical research to inject humans with a specific antigen for non-therapeutic purposes. And to perform a partial splenectomy to collect antibody-producing cells. Therefore, it has been necessary to devise other approaches for obtaining human monoclonal antibodies. To address this need, researchers constructed a xenomouse in which 1. The mouse antibody production machinery was inactivated, and 2. All of the human immunoglobulin loci, both light and heavy chains, are integrated into a mouse chromosome. The human heavy chain genes and the human K and lambda light chain genes, where K and lambda are different classes of light chain genes, were cloned onto yeast artificial chromosomes, YACs that can carry very large amounts of foreign DNA. The viruses with human immunoglobulin genes were then introduced into mouse embryonic stem cells by fusing YACs, C containing, that is yeast artificial chromosome containing yeast spheroblast, with the embryonic stem cells. This procedure yields a large number of embryonic stem cells in which all of the introduced human immunoglobulin genes have become stably integrated into the chromosomal DNA. These transfected cells were used to generate mice containing human immunoglobulin gene loci, crossbreeding of two mouse lines, one carrying both mouse and human immunoglobulin genes, and the other carrying the deleted mouse immunoglobulin genes, produced a mouse strain called Xenomouse that expresses only human immunoglobulins. It is not possible after immunization of a Xenomouse with a particular antigen to produce a fully human immunoglobulin. The large human antibody repertoire and the Xenomouse has enabled researchers to produce a number of fully human antibodies, many of which are currently at various stages of clinical development. For example, the first fully human monoclonal antibody produced using this technology is panitumumab, which was approved in September 2006 for treatment of certain forms of colorectal cancers. Next we go to is antibody fragments. At the end of the lecture or start of the next, as a separate lecture, I'll give you the keywords for each chapter completed so that you know where to start and where to stop. Right? So antibody fragments. Naturally occurring antibodies are highly specific targeting reagents. They provide animals with a powerful means of defending themselves against a wide range of pathogenic organisms and toxins. Immunoglobulin G, IgG, is the main antibody found in mammalian serum. And it is the native form that is the almost exclusively used in therapeutic antibodies. The fact that IgE molecules have two identical sites that bind to two identical antigens, that is, they are bivalent and generally increases the effectiveness in vivo. While the Fc portion of the Ig molecule is important in recruiting cytotoxic effector functions through a complement or interaction with specific receptors, Fc-mediated effects are not necessary for all applications and may even sometimes be undesirable. Over the past several years, by manipulating portions of the IgG light and heavy chain cDNAs, researchers have constructed a variety of IgG derivatives or fragments that can be used instead of whole body antibody molecules. Some of these molecules, because of their small size, bind more effectively to targets that are inaccessible to conventional whole antibodies. Others have multiple sites for binding to the same antigen, while others have binding specificities for two or more large antigens. Initially based on Fab and Fe fragments of antibodies, antigen binding single protein chains, SCFE, consisting of only VL and VH domains, were developed. Single chain antibodies may be used for a variety of therapeutic and diagnostic applications in which Fc effector functions are not required and when small size is an advantage. Now, single chain antibodies have a molecular mass of approximately 27 kilodalton compared with approximately 150 kilodalton for Ig molecules. Due to their small size, single chain antibodies can penetrate and distribute in large tumors more readily than intact antibodies. This lecture will be over in a minute and a half from now, and the rest of the portions will be continued in the next lecture. Until then, let me just sum up with a few more lines. Right? In addition, a protein coding sequence can be linked to a single chain antibody sequence to create a dual function molecule that can both bind to a specific target and deliver a toxin or some other specific activity to a cell. The computer simulations of 3D structures of potential single-chain antibodies show the V, L, and VH domains have to be separated by a linker peptide to assume the correct conformation for antigen binding. And on the basis of this design constraint, DNA constructs the VL and VH sequences from a cDNA of a cloned monoclonal antibody where each ligated to chemically synthesize DNA linker fragment in the order VL linker VH. And after expression in E. coli, the single chain protein was purified, and both its affinity and specificity were found to be equivalent to those of the original intact monoclonal antibody. The effectiveness of this disulfide stabilized Fe molecule, VLSSVH coupled to a cancer cell toxin, was compared with that of an SCVF molecule coupled to the same toxin. So the disulfide stabilized an SCV immunotoxins, the same activity and specificity, and the shortening, the link affects not only demultimerization but also the stability of the molecule with